ವಿದ್ಯಾರ್ಥಿಗಳು ಕಳೆದ ವರ್ಗದಲ್ಲಿ ನಾವು ಸ್ಪರ್ಮ್ಯಾಟೋಜೆನೆಸಿಸ್ ಅನ್ನು ಕಂಪ್ಲೀಟ್ ಮಾಡಿದ್ದೇವೆ. And in this class we are going to be starting oogenesis. What is meant by oogenesis? It is very clear. You know that it is a formation of egg. The process of formation of egg is said to be oogenesis. Yes, it's a very simple definition. When compared to the spermatogenesis, it is totally different. We said that already. The spermatogenesis is the spontaneous one, but oogenesis is not as spontaneous because it starts in the embryonic stage only. The process of the mature ovum, that means starts from the embryonic stage. See, in each fetal, the development of egg starts there only. At that stage, each ovary of that fetal may have 2 million follicles. But at the time of delivery, we are going to get in, so that means there is a slowly degeneration of follicles takes place. Anyway, oogenesis starts in the embryonic stage, that is in the ovary of embryo of that female one. Oogonia, we are going to be saying, it is known as an embryonic mother cell. Already it is clear that no more oogonia are formed after the birth. Oogonia are going to be multiplying, and there is a formation of primary oocyte. This primary oocyte enters the first meiotic stage, that is the prophase 1, and get arrested. Allige stop agutte. And afterwards ovulation time alli continue agudaru nodutte. You observing here the different structures are there. Primary oocyte, that is primary follicle, secondary follicle, tertiary follicle, and the Graafian follicle. At the beginning, the primary oocyte, it is get covered by the new cells. We are saying granulosa cells. And after that, this primary follicle get turned into secondary follicle, and we are observing still more new cells are formed. We are saying these granulosa cells are covered by the theca cells, and in the tertiary follicle, these theca cells are get converting into theca interna and theca externa. And the granulosa forms here, there is a formation of cavity. We are saying antrum. Antrum anta ಹೇಳ್ತಾ ಇದ್ದೇವೆ. This oocyte is covered by the cells, new cells. We are saying zona pellucida. Zona pellucida is a new layer is formed around the oocyte. Granulosa cells are there, and at the time of these tertiary follicles are get converting into the Graafian follicles, there is a formation of cumulus operus, corona radiata, granulosa cells, antrum cavity, then theca externa and theca interna are formed here. that is the mature Graafian follicle. See, I already said that many primary follicles are degenerate during the phase that is birth to the puberty, attains the puberty of the female. After puberty, we are going to get in that only the 60 to 80,000 follicles are left. Remember, each egg is released in 1 month, that is for 28 days. In the tertiary follicles, the primary oocyte enters the first unequal meiotic division and forms the haploid secondary oocyte. And one more most interesting here, the first polar body is formed. Not clear about the polar body, whether it degenerates. But the secondary oocyte now retains the very rich nutrients, the cytoplasma enriched nutrients, because that is going to be entering for the fertilization. This is secondary oocyte, I already said here, it forms the layer of cells we say called as the zona pellucida. This is secondary follicle get converted into tertiary and afterwards the Graafian follicle, and it is now ready for, it is a mature one, it is ready for the ovulation. This is the whole process of oogenesis takes place. Oogenesis, in the examination they are asking that schematic representation. I will go for that schematic representation. Yes, students, now you are observing here the schematic representation of the oogenesis. This one is going to be asking in your examination. Please concentrate here. You are observing that here the first one is that is, you are observing the diploid oogonia, two mother cells are present. Here it is all everything is going to be taken in fetal life, that is up to the birth. That is multiplication, growth of that oogonia, everything. And from birth to puberty, you know that so many follicles are degenerate, and after birth only we are going to getting 60 to 80,000, and no more extra oogonia are forming after the birth. Then in the puberty, so we are observing that after the maturation, slowly the cycle starts, and we are observing that there is a formation of first polar body and the second polar bodies are going to be forming. That means the first meiotic division and second meiotic divisions may occur after that. So we are start here the oogonia. It increases its number in ovary by the mitosis and undergoes the process of differentiation. The primary oocyte is formed, that is before the birth. Okay, the primary oocyte is formed and starts the first meiotic division. First meiotic division ಅಲ್ಲೇ ಫಿಟಲ್ ಸ್ಟೇಜ್ ಅಲ್ಲಿ ನಾವು ನೋಡ್ತಾ ಇದ್ದೀವಿ, and later on it ಅರೆಸ್ಟ್ ಆಗುವುದನ್ನು ನಾವು ನೋಡಿದ್ದೇವೆ. The first meiotic division completed prior to the ovulation. ಎಲ್ಲಿವರೆಗೆ ಫಸ್ಟ್ meiotic division ಕಂಪ್ಲೀಟ್ ಆಗುವುದಿಲ್ಲ ಅಂತಂದ್ರೆ ಓವ್ಯುಲೇಷನ್ ಹಂತದವರೆಗೂ ಈ ಫಸ್ಟ್ meiotic division ಕಂಪ್ಲೀಟ್ ಆಗುವುದಿಲ್ಲ. Before the ovulation, this first meiotic cell division completes. And what about the second meiotic division? That means the second phase of meiosis 2. Idu yavaga agtade? See, when the sperm touches that zona pellucida and the membrane of that induces the completion of the meiotic cell division. That is the thing here. Okay? Completion of the secondary, that means second meiotic cell division. Yes, the primary oocyte undergoes the first meiotic division and there is a formation of first polar body, and afterwards the second meiotic cell division takes place, and there is a formation of second polar body, and we are going to be getting that only the functional ovum. But in spermatogenesis we have seen that all cells, 4 cells are the functional, but here only one becomes a functional one. That is the most important one, and that is the difference between the spermatogenesis and oogenesis. Okay, I hope it is clear for you. The secondary oocyte, it is get converting into ovum. Okay, and now here you are observing that the structure of egg. It is very clear that ovum is there. It is covered by the perivitelline space is there. There is a small gap, and it is covered by the zona pellucida, and this zona pellucida is covered by the cells of corona radiate. Okay, this zona pellucida is the one after the entry of the sperm, it blocks, it is get converting, it cannot allows, it blocks the entry of additional sperms. When the sperm touches, okay, so that induces the completion of the second meiotic cell division. Okay, this is about the oogenesis and the structure of the ovum. I hope it is clear for you. Go through the textbook. I am very brief. What is there in the textbook, that much only I am covering, because you are already observing the more video clips, and my video clips also. Better you go through the textbook, that becomes clear. If you have any doubts, you can ask whether the Kiran sir or for me. Okay.